Hello and thank you for joining this talk. So my name is Magnus Jacobsen and I work at situ in Denmark. This talk is about our work on a so-called environmental sample processor that can be used for autonomous sampling of eDNA. So eDNA is an alternative to traditional monitoring methods. This is also true for offshore monitoring, where eDNA may prove a cheap alternative. However, sampling is still expensive and it relies on boat time, often from dedicated research or fishing vessels. The associated cost may reduce sampling efforts to single snapshots, which may not be adequately representative of whole biodiversity. There may also be a time lag from sampling to results, which may hinder fast response time that could be important if you work on, for instance, invasive species. One way to bypass these problems is to use an automatic sampler. One such sampler is a second-generation environmental sample processor, or 2G ESP. This is an instrument that can conduct automatic water sampling, filtration, and eDNA extraction. The extract can be analyzed by up to 6 species-specific qPCR assays. It can also archive filters for post-deployment analysis in the lab. During deployment, it's connected to a telebuoy, which allows direct streaming of the results in real time, and also communication between the ESP and the end user, so the end user can change sampling protocols during deployment. The ESP is powered by batteries, which allows up to several months of sampling depending on sampling effort. In this figure, in the top right corner, you can see the ESP without its underwater housing, and in the figure below, you can see a drawing of the mooring where the ESP is deployed pelagically with the telebuoy in the surface. The ESP can be deployed in many different ways. In the top left corner, you can see the ESP deployed dockside. Here it's connected to a pump that pumps water into the ESP. To the right, you can see the orange kind of a pelagic module, which allows the ESP to be buoyant and placed in the middle of the water column during deployment. And below, you can see the ESP attached on top of this landing module, which allows us for seabed deployment. During deployment, the ESP processes samples in a step-to-step kind of way. So for the in situ analysis, we'll first filter the water, and it would lyse the filters and purify the DNA. Afterwards, then we'll analyze the DNA with each assay one at a time. For the archival sampling, it will filter the sample, filter the water, and then it'll add preservatives to the filters and store them for post-deployment analysis in the lab. We had several problems along our way. So there's been different problems with equipment malfunction, but the biggest problem has probably been inhibition of our reactions. This has been due to the master mix we used, which worked really well on the ESP, but not so well for the environmental samples. These are problems we dealt with, and we now stand with kind of a really well functioning ESP. There are still some problems. One of them is that we find lower sensitivity of the in situ analysis compared to the archival samples. This is probably due to the analytical pipeline and the modules used on the ESP, which is something that we are looking into. So on this slide, I just want to show you a few results from our deployments. This is from a deployment from 2020, where we looked into different invasive species in Denmark. So these are analysis of qPCR from the archival samples, and it shows the number of DNA copies per liter on the y-axis. On the x-axis, we have the dates, which covers basically 2 weeks of deployment. What you can see is that the number of DNA copies fluctuates a lot in both species, basically going from thousands of copies to none. Also, you see that there is a difference when we find high and low levels across species. So there's both a temporal difference, but also a species difference. This is very interesting and something that we're investigating at the moment. So how does this relate to oceanographic changes, like for instance to changes in sea currents and to biological differences? Also, we are analyzing these samples not only with qPCR but also with metabarcoding approaches, which allows us to analyze many more species in the same time. Before we end, I'd like to thank some people that helped us along the project. First of all, I'd like to thank Mbari for always being there and helping us with all kind of issues on the ESP. Without them, it has definitely not been possible. I'd also like to thank DHI, which are one of our main collaborators on the project, along with many other people helping us along the way and involved in various projects on the ESP. If you're interested in our work or have any questions to our work, feel free to contact us on any of the emails here below. Thank you.